Hey guys, welcome to my channel. So basically I'll be answering questions and giving you the best answers with some tips and tricks as well for the Cambridge International Examination or CIE A level biology syllabus, and this is for the chapter number 15, also known as control and coordination. So let's get started. Okay, first one. Most reflex arcs pass through the spinal cord and involve different types of neurons. Name and state the functions of the three types of neuron in a spinal reflex arc. So, guys, if you know or if you can recall a reflex arc, what is a reflex arc? Is basically a pathway along which electrical impulses or action potentials travel to bring about a reflex action without involving conscious regions of the brain. So yeah, just a heads up. So as we all know, there are three types of neurons. Well, in this A-level biology syllabus, basically we have sensory neuron, we have relay neuron, and we have the motor neuron. So basically I've drawn a diagram for the sensory neuron so you can have a better understanding. Basically, in a sensory neuron, you have your Schwann cells which wrap around the axon and form the myelin sheet. So yeah, you have the axon here. This is your cell body, and this is your dendrite or many dendrites, and this is your axon terminals. So yeah, this is for the structure for sensory neuron. And so what I've written is a sensory neuron, basically a neuron which carries electrical impulse from the receptor to the central nervous system. And then after that we have a relay neuron, also known as an intermediate neuron. It's found within the CNS, and it carries electrical impulse from the sensory neuron to the motor neuron, and the motor neuron carries electrical impulse from the CNS to the effector, which could be a muscle or a gland. And I have also drawn the structure of a motor neuron. So as you can see, a motor neuron also has a cell body, dendrites, axon terminals, axon itself, myelin sheet, again made up of Schwann cells which wrap around the axon. So yeah, that's it for this part. So let's move on to the next. So then we have the B part. Some poisons affect the functioning of synapses between neurons. Yes, listed below are four organisms A, B, C, D, each of which produces a different poison that affects the functioning of synapses. Okay, so basically guys, if you remember, what is a synapse? It is a junction between two neurons. Okay, so I repeat again, a synapse is a junction between two neurons. Okay, so then we have A, B, C, D. These are four organisms producing poisons which affect the functioning of synapses. So as you can see in table 7.1, A blocks sodium channels, B blocks calcium channels, C prevents exocytosis, and D inhibits acetylcholine esterase. So we'll move on to the next question, which is basically they're asking suggest and explain how poison from each of these organisms affects the functioning of synapses. So if you can recall from the organism A, it affects the sodium channels, right? Blocks sodium channels. So when your sodium channels are blocked in a synapse, what happens? Sodium ions cannot diffuse into the postsynaptic neuron. So what happens when sodium ions cannot diffuse into the postsynaptic neuron? There will be no depolarization, and if there is no depolarization, no action potential can be generated in the postsynaptic neuron. Okay, and then the organism B, it blocks calcium channels. So when calcium channels are blocked, calcium ions again cannot diffuse into the presynaptic neuron. So if calcium ions cannot diffuse in the presynaptic neuron, the vesicles inside the or found in the presynaptic neuron that they contain vesicles. So and these vesicles contain neurotransmitters. So if calcium ions cannot diffuse into the presynaptic neuron, these vesicles containing neurotransmitters cannot move towards and fuse with the presynaptic membrane. And C, organism prevents exocytosis. So if there's no exocytosis, acetylcholine cannot be released into the synaptic cleft. And then organism D inhibits acetylcholine esterase. So guys, if you can recall or remember, uh, acetylcholine esterase, it's an enzyme which is found in the synaptic cleft, and it breaks down or hydrolyzes acetylcholine into acetate and choline, and after a few milliseconds of its release in the synaptic cleft, acetylcholine is broken down or hydrolyzed by acetylcholine esterase in order to prevent continuous firing of action potentials in the postsynaptic neuron. So this is basically what I've written, and yeah, that's it for this question. So we will move on to the next one, which is, yes, this one for muscle contraction. So figure 3.1 shows a diagram of two sarcomeres of relaxed striated muscle. Guys, striated muscle basically comes from the word like, stripes. So this muscle has a lot of stripes, as you can see. Like, a zebra has a lot of stripes. So, this is also having lots of stripes. So that's a heads up if you don't know. So we can see we have the H zone, the I band, the A band, and the Z line. So I don't want to go into too much detail as we're just solving papers. So when the striated muscle contracts, state what happens to the length of the I band. So guys, always remember, when a muscle contracts or there is contraction, the I band always shortens. Okay, always I band always shortens, and the A band, when muscle contraction occurs, A band's length will not change. It's as simple as that. So moving on to the next one, it says describe how the response of the sarcoplasmic reticulum to the arrival of an action potential leads to the contraction of striated muscle. Okay guys, whenever you see this word contraction, always think of depolarization or depolarizes. So basically what you can write is that the membrane of the sarcoplasmic reticulum depolarizes. So when depolarization occurs, this will cause voltage gated calcium channels in the membrane of the sarcoplasmic to open, causing calcium ions or an influx of calcium ions out of, it will cause calcium ions to diffuse out of the sarcoplasmic reticulum and into the sarcoplasm. So now the calcium ions, they will bind to the troponin present on the thin filament, and troponin will change shape and will pull away tropomyosin, and it uncovers or exposes myosin binding site on actin, and because of this the myosin heads bind to actin, forming power strokes. So this is how basically contraction of striated muscle happens. So moving on to the next one. So this is for the plant growth hormones. So gibberellin is a plant growth hormone that has a role in germination. So basically germination means to grow. So yeah. And in stem elongation, outline how gibberellin is involved in activating genes for stem elongation. Okay, so basically guys, gibberellin, so it's a hormone. We all know hormones have their respective receptors. So gibberellin, it will bind to the receptors on the cell surface membranes of cells of aleuron layer. So when binding will occur, receptor changes shape, and then when receptor changes shape, it will result in the activation of an enzyme. So as we all know, this enzyme will break down DELA proteins, and when DELA proteins are broken down, PIF is no more blocked by DELA proteins, and PIF binds to the promoter of amylase gene. And then when promoter binds to his gene, transcription of amylase gene begins. So yeah, that's it. You will score 2 marks if you write all this. And then the B part, it says outline the role played by gibberellin in the growth, or we can say germination, of wheat seeds. So basically, the gibberellin, it has embryo. So these cells of the embryo, no, I'm sorry, the cells of the embryo, they will secrete gibberellins, and then these gibberellins will diffuse from cell to reach the alluron layer, and then transcription of amylase gene begins, mRNA is produced, and then so on and so forth. You can just take a screenshot and write all this. So basically, yeah, that's it. And then moving on to the next part. Okay, and yeah, I think this will be the last one. Yes. Okay, so basically they're asking explain what is meant by the term voltage gated channel. Okay, so basically guys, a voltage gated channel, it's a transmembrane protein which opens or closes to allow transport of ions. If you write this, you can score 2 full marks. So moving on to the next. So C part is asking explain the role of acetylcholine esterase in a synapse. So yeah, as we discussed, or as I said previously, acetylcholine esterase is an enzyme found in the synaptic cleft. It breaks down acetylcholine into acetate and choline, and then after a few milliseconds, when after it is released in the synaptic cleft, acetylcholine is then broken down by acetylcholine esterase to prevent continuous firing of action potentials in the postsynaptic neuron. So yeah, you can write this. And then the last part is outline the roles of synapses in the nervous system. Okay, so this question is for 2 marks, but I've given you one extra one so you can remember it. So basically, the role of synapses in the nervous system are this, which is to ensure one-way transmission of action potentials. And then the second one is synapses are involved in memory and learning. And then last but not least, synapses also allow interconnection of nerve pathways to provide a wide range of response. So yeah guys, so basically these are the questions and answers related to chapter number 15 of the CIE A-level biology syllabus. So I hope this video so I hope you found this video helpful, and if you do, please like, share and subscribe. If you have any questions, you can post them down in the comments below, and I will be happy to respond. So yeah, I will just go back up so you can take a screenshot. So yes, I'll just zoom out for you guys or whatever.